I know it's very.
Okay. We'll give people a couple of more minutes to trickle in. Because you could now go there. You could go to your opening slide.
It's a general title. That's what I said. I just put on a general thing. Yeah. But you're puzzled looking at the...
No, that's okay. All right, why don't we just go ahead and get started as I think people continue trickling in. It's a real pleasure for me to introduce Erik Procko. Erik and I, we finished PhD about the same time, 2009, 10-ish, and then he got his PhD at Harvard, and he was heading to David Baker's lab for a postdoc, and I was finishing my PhD in David Baker's lab and heading to Harvard for a postdoc. So we literally crossed paths going in opposite directions. Erik's lab, he'll describe what he does with, high throughput measurements for protein sequence functional landscapes. But what his work really caught my attention. This was, just as the pandemic was starting. So this is March 2020 or thereabouts when, the university was shutting down, and all of us were home. And so in my lab Slack channels, we'd be reviewing, old papers in coronaviruses and maybe some of the more recent ones in SARS-CoV-2. And so this is March 2020. And, we were discussing ways in which we could design, potential inhibitors against SARS-CoV-2. And so, one discussion point that came up was, it'd be nice if there are some measurements on the ACE2 protein receptor that SARS binds to, and there are functional measurements if someone had made. It'd be really cool because then we could use that information to design inhibitors against SARS-CoV-2. And so this was a discussion that was happening in my lab Slack channel. And by end of March 2020, there was a paper on bioRxiv where someone had done a complete mutational scan of the ACE2 receptor and put it on bioRxiv. I was really sort of stuck by that because, we'd known that the virus was getting started like in February 2020. And by the end of March, there was a full manuscript on this, just, it's 3 years of work. And I was like, how the hell could someone do this? And anyways, but Erik had done that. And since then, he started a company, and he's sort of taken this technology forward to build these decoy receptors, which are mutant versions of ACE2 that could soak up SARS-CoV-2. And so I'm looking forward to Erik's work. Listening about it. Thank you.
Thank you. This is on. You can all hear me? Perfect. Okay. So good afternoon. The work I'm going to talk about today is to align with a lot of the interests here at Wisconsin. I'm going to talk about some of our work on engineering and understanding the mechanism of action of a particular couple of chaperones. And all of this work comes from my academic lab at the University of Illinois. As Vatsan mentioned, I ended up founding a small startup company primarily based on our work on SARS-CoV-2, but also some earlier work on other viruses. This image at the background here is an example of that, of a viral spike binding to its receptor, where we make some mutations, where we can introduce specificity and affinity. And I founded a small startup company, and merged with a slightly bigger small startup company called Cyrus Biotechnology. And I still have an appointment at Illinois. I still have a few students there, but most of my day-to-day activities are now with Cyrus Biotechnology. I have just one slide in here which shows how things I've learned and study in the academic lab are very relevant for biotech as well, if I get there at the end. But otherwise, yes, all from Illinois here. Okay, so the kinds of questions that we have been interested in my lab are how does protein sequence dictate shape and conformation in dynamic membrane proteins? And the bigger the motions, the more interesting they are. So this really sent us looking at proteins in neuroscience, certain receptors, transporters, as well as it's what led us to looking at virus host interactions, in particular the conformational changes that happen for membrane fusion proteins that mediate infection. And then what can we learn about membrane protein mechanism from mutational landscapes? And these mutational landscapes are experimental in nature, where we use big data methods to look at many thousands of mutations. And then finally, how can we leverage that information for engineering proteins with biomedical applications? And so my group has studied a number of different topics, all related around membrane proteins and especially dynamic membrane proteins. But the work I'm going to talk about today involves antigen presentation in the immune system. So inside all of your nucleated cells, proteins are getting ubiquitinated, and then they're getting shuttled to the proteasome, where they are degraded into small peptides for their turnover. And some of those peptides get moved through a transporter into the endoplasmic reticulum. And then in the endoplasmic reticulum, these peptides here can be loaded onto these presentation molecules called MHC class 1. In humans, MHC is known as HLA, human leukocyte antigen. So you may find in this presentation I switch between saying MHC or HLA. For most purposes, it's the same thing for today's talk. So these peptides get placed onto MHC. When that happens, these MHC molecules shed chaperones that they are bound to and are keeping them in the endoplasmic reticulum, and then they can migrate through the secretory pathway to the cell surface, where that peptide is now presented to a T cell. And if there is a high affinity interaction here, then that would mean that the peptide here is abnormal in some way, maybe cancer associated, maybe from a virus, and then that will be the cue that tells the T cell that there's something wrong with this cell and it's going to get targeted for death. There is a lot of diversity in this system, a lot of polymorphisms. So there is diversity at the level of the T cell receptor, many millions of different T cell receptor specificities in the body. But there's also a lot of diversity in different MHC alleles. And that's important because you don't want the entire human population to have all of their eggs in one basket. And these MHC alleles have some specificity for different peptides. So by having allelic diversity, different people will present different kinds of peptides. So there's some sequence patterns that these MHC alleles look for. So each of us has a number of different alleles, and in the human population there are around 3,000 total, and each allele can present around 10,000 different peptides based on the motif that it recognizes. So here's what an MHC class 1 molecule looks like from what the T cell is seeing. So this is kind of a face-down view. There's an antigenic peptide derived from the proteome inside of the cell. That's being displayed here in orange, and it's being held in a peptide binding groove or cavity. So there's a beta sheet at the bottom, and then there's these alpha helices that rim that cavity, and then certain residues of the peptide are pointing down into pockets. They're being clasped. That's what's giving the MHC1 allele its unique specificity for certain peptide sequences, but then the other residues are pointing out into solvent. They can be anything, and that's what the T cell receptor is coming along to look at and interrogate. So here it is again, some kind of antigen presenting cell presenting on a class 1 MHC, a peptide shown here in red, and then a T cell receptor is recognizing that combination of an antigenic peptide and the MHC molecule that is presenting it. This is what the structure looks like. The MHC molecule here has a heavy chain shown in gray, and then it is non-covalently associated with a second subunit called beta-2 microglobulin. So we have a beta-2 microglobulin with a heavy chain, and then here's the peptide binding cleft here with a peptide shown in red. And then getting that peptide loaded onto this MHC requires a whole lot of chaperones inside of the endoplasmic reticulum, a complex forms. It's known as the peptide loading complex. There are housekeeping chaperones such as calreticulin and ERP57, but the most interesting chaperones for people studying antigen presentation are two chaperones which are solely dedicated for class 1 loading. So these are class 1 specific chaperones, and they are tapicin, shown here in orange, and a second chaperone called TAP-BPR. These two chaperones are structurally almost identical. There's some small differences. They do have slightly different roles inside of the cell. Tapicin is the most important of the chaperones. If you knock it out, a lot of MHCs never get loaded with peptide. They therefore stay inside of the cell and they never make it to the cell surface. TAP-BPR is not critical. It has more of a quality control process in the Golgi, but you can functionally replace tapicin in tapicin knockout cells with TAP-BPR. I'll show data where we did that exact experiment. And we do most of our experiments with this second chaperone called TAP-BPR, simply because tapicin is a pain to purify. It forms a disulfide bond with one of the other chaperones here, not easy to work with. So we use TAP-BPR as a model system, but we extrapolate our findings to both of these chaperones. So here's when we started the project, this was the idea of how these chaperones work. So here would be an empty MHC. So it doesn't have a peptide bound, it's unstable, and it is held in a peptide receptive conformation through interactions with this class 1 specific chaperone, tapicin or TAP-BPR, shown in orange here. And then if a high affinity peptide comes in, it changes the conformation of the MHC as it clasps onto that peptide. That reduces the affinity for the chaperone. The chaperone is released. This peptide MHC then moves on to the cell surface to present the antigen to a T cell. So we began by looking at the different alleles that interact with our model chaperone TAP-BPR. So we looked at 4 different class 1 alleles here. So we have what's known as an A1 allele, A2, HLA-A2. These are 2 human alleles. We also looked at 2 mouse alleles just in this panel here. Everything's binding to human TAP-BPR. And this is size exclusion chromatography. So if the 2 proteins don't interact, we see 2 independent peaks. And when they do interact, we see a leftward shifted single peak showing that they're forming a nice co-eluting species. So the point here is that a couple alleles here did not interact by size exclusion chromatography. A couple do, so there are differences. And then we could also look at it by NMR. Size exclusion chromatography is not an equilibrium measurement. As 2 proteins dissociate from each other, they will now separate out on the gel filtration matrix. NMR is an equilibrium experiment. It can give some more quantitative numbers. Here, I should also mention, this is work from Nick Skarakis' lab. So this was a very close collaboration between our 2 groups. He's an NMR spectroscopist, actually came from David Baker's group as well, developed CS Rosetta, very popular in the NMR community. And he was studying these proteins, but wanted to move beyond just a method of applying NMR, really trying to branch out to other biochemical assessments and cell-based assays, and that's where we teamed up together. So whenever you see NMR data, that's from his group. Whenever you see cell-based data, that's from my group, and then some biochemical assays from both of our labs here. So that's really very much a joint effort, all of this work. So they labeled the methyl groups. So they made the class 1 MHC, they refolded it from E. coli, labeled different methyl groups on the aliphatic side chains, and then shown here, for example, let's take this example here of HLA-A2. Shown in gray is the NMR spectrum in the absence of the chaperone. You now throw the chaperone molecule in and you remeasure the spectrum. That's shown in blue here. And if there's an interaction between the two proteins, then those methyl groups, those labeled methyl groups that are at the interface, are now in a new chemical environment, and therefore they're going to have a change in their resonating frequency. So we look for these changes in chemical shift. So this example here in the center for HLA-A2, this is an example where we didn't pick up an interaction by size exclusion chromatography, but we do pick up an interaction by NMR. We're seeing shifts here, excellent interactions with these two murine MHC1 alleles, no interaction here with HLA-A1. So we have different alleles with different propensities to associate with the chaperone tap BPR in vitro. Went further here, these are CPMG relaxation dispersion experiments where you can get information on dynamics on the micro to millisecond time scale here. And these little spheres, these each are for a single labeled methyl group, for a single isotope label here. And the color indicates the degree of dynamic motion taking place. So gray would mean that it's pretty static and rigid on the time scale of this experiment, whereas as you go to warm colors up here, then there's a lot more dynamic motion going on. And what we observed is that for a non-interacting allele, HLA-A1 in orange, there's a lot of gray here, not a lot of motion happening in this molecule. But as you move towards the alleles that are binding to tap BPR, the chaperone, very well, you're seeing a lot of green, some red and yellow here. So there's a correlation between dynamic motions of the substrate and its binding to the chaperone tap BPR, which is summarized just here. So to validate that, a disulfide staple was placed into the MHC1 molecule. So this is where that disulfide bond was introduced. The crystal structure for the disulfide bonded form shows excellent electron density for where the disulfide is placed. And then look to see, well, do you still bind to the chaperone? So again, the labels are on MHC class 1. You add in tap BPR and you look to see, are there changes in the chemical shifts indicating a physical interaction? So we see that there is an interaction between the wild type MHC1 allele and tap BPR. So the gray is shifting to the blue here. But now when you put in the disulfide staple, the gray and the orange spectra are perfectly overlaid. So there's no interaction here, or very weak interaction. And then if you add a reducing agent, TSEP, to break that disulfide staple, now you regain the interaction. So this all looked very nice, and we wanted to confirm that this is also true inside of cells. So the experiment we turned to is bimolecular fluorescence complementation, also called BIFC, or some people call it BIFC, where we took Venus, which is a derivative of yellow fluorescent protein, and it's split into an N-terminal half called VN and a C-terminal half VC. MHC1 and tap BPR, these are both transmembrane proteins. They both have a single transmembrane helix. And on the cytosolic side, so this would be up in the endoplasmic reticulum here, on the cytosolic side, we place VN on the MHC, we place VC on tap BPR, and if they form a physical association, then you get complementation and you observe yellow fluorescence. So we did that in cells. CXCR4, it's an unrelated chemokine receptor, it's a negative control. So we're looking for complementation, BIFC, between these proteins here and TAP BPR. CXCR4 is a negative control. We look at our MHC1 allele. We saw a very nice biFC signal indicating a good interaction inside of the cell. You put in the 2 cysteines to try to form that disulfide staple, and the prediction was it should be less, there shouldn't be an interaction, and not only was it not less, if anything, it was more. And it got worse when we studied it further. We looked at a whole bunch of different HLA alleles where we knew that some of these do not interact with the chaperone TAP BPR in vitro. Put them all into cells, measure biFC, we're picking up interactions everywhere. We know it's not an artifact because we have another assay that we can do, which is where in wild type cells, which already have tapicin and TAP BPR present, and if you now add an overexpressed amount of TAP BPR, too much of the chaperone is going to keep those MHC molecules inside of the cell. They interact in the ER, and it's retaining them, and their localization to the surface is retarded. So we can look at all of these different alleles going to the cell surface, that's shown in white, and then in the gray bars, when you overexpress TAP BPR, you're now keeping them inside of the cells, so they are all interacting. So this was a problem, and it required us to reimagine what was actually happening inside of the cell versus what was happening in the in vitro experiments. And what is happening inside of the cell, of course, is that in the endoplasmic reticulum, you don't have beautiful folded proteins like you have in vitro interacting with the chaperone. You have a nascent MHC1 that has just come off the ribosome, just gone through the translocon, spat out into the ER, and it's not yet fully folded. And it's bound to all of these other chaperones as well. So you've got this complex here. So the idea was that maybe we have broad recognition of MHC molecules when they're in their nascent form, but as they mature and they become more rigid and have less dynamic motions, then the interactions with the chaperones start to become more restricted. And if we could look at the cell surface, presumably most of the MHC that goes to the cell surface is bound to a peptide and is properly folded and now mature. If we could localize the chaperone to the cell surface, then now we might have something that's more akin to the in vitro experiment. And so we did that by replacing the transmembrane and cytosolic tail of TAP BPR with a canonical transmembrane helix. It goes to the surface very nicely. A lot of it is still intracellular, but there's also a very large amount of the cell surface, worked very well. And then now what do we see? Now we see, compared to the wild type, when we put in the 2 cysteine residues, we do indeed knock down the interaction. So we took this further by wanting to say, well, what are the key attributes of the MHC then that are being recognized by the chaperone? And we looked at that through deep mutagenesis. So in my group, we put in a lot of effort to optimize methods for deep mutagenesis in mammalian cells. Our methods are a little different than what other labs in the field do, but they have pluses and minuses. And the idea is that we transfect cells under conditions where you typically get only one gene copy of our gene of interest per cell, no more than one. And then we can do some kind of selection on this library. So this is showing an example of cells expressing different receptors. And we come along with a fluorescent ligand in red here. Some of them bind to a lot of it. Some of them bind to not very much. Some not at all. So they have different fluorescent properties on them. And we can use FACS sorting to pull out the cells of interest. I'm going to show you this kind of experiment where we use biFC as the selection. So we could pull out cells that were more fluorescent than other cells based on interactions inside of the cell between an MHC substrate and TAP BPR. So after we sorted the cells, we deep sequence everything. We deep sequence what's in the initial library. We deep sequence what comes out at the end. And then we can determine which mutations are enriched and depleted and calculate a mutational landscape. So here's what the data looked like for MHC1. So we did a mutational scan on the peptide binding groove domain of MHC1. And we did a selection for surface expression. So you don't get to the, and we did this on HLA-A2. So this is an allele that we know can interact with TAP-BPR both inside of a cell and in vitro. It's also a very important human allele in many diseases. So we looked at surface expression. This tells you something about binding to a peptide and folding. You don't get to the surface if you're not folded and bound to a peptide. And then we also did a selection for BIFC between HLA-A2 and TAP-BPR. Orange mutations, orange boxes mean a mutation was bad and depleted. White means it was neutral, and then dark blue would mean that it was enriched. And you can clearly see that these are very different, and that where there's a lot of orange up here, a lot of those mutations are allowed for binding to the chaperone, even though almost certainly many of those mutations you would anticipate will be destroying three-dimensional shape and structure. So this is mapping conservation scores. So we just average all of the log2 enrichment ratios for each position. So orange here would mean that that position's highly conserved. Pale colors would mean it's reasonably tolerant of mutations. If it gets very blue, then that would mean that it's actually a hot spot for enriched mutations for change. And you can see that for surface expression, there's a lot of orange here at certain pockets of the peptide. These are the A and the B and the F pockets here. These are the pockets where you clamp onto the amino acid side chains of a peptide. So this immediately indicates that surface expression, you must have properly formed pockets for binding and presenting a peptide antigen, exactly as we would expect. But for binding to the chaperone, shown here in green from this crystal structure, then you can see that, well, there's not a lot of orange. There may be some here, but not a lot over here. You can really have a whole lot of mutations. Does anything go? Well, not quite everything. If you do a cross-section through here, what you see is that in this region here, there is an orange patch where there's mutational intolerance. So there is an element of structure, a substructure that must form in this region to get productive binding to the chaperone, but otherwise a whole lot of the rest of the protein can be changed. And we put in some targeted mutations. We chose prolines because prolines are frequently highly disruptive to secondary structural elements. So we put in some prolines on this helix here. We put in some prolines over on this side here. This is showing the BIFC up the top. So if you look at the BIFC, all of these prolines here, they're color-coded by their BIFC signal. These ones are well tolerated, and you still have very nice interactions between the chaperone and the MHC. But when you start putting some prolines in over here, not so good. And we also looked at surface localization, where here, so in white, the white bars are when we just put in the mutated MHC into cells, and we see that all of these proline mutants are not getting to the cell surface. That immediately tells us the proline mutations are doing exactly what we expect. They are destroying the structure, and it's no longer properly folded, doesn't make it to the cell surface. But if you add in our TAP-BPR that goes to the cell surface, this TAP-BPR can bind to these different misfolded MHCs to various extents. It can bind to them, and it can then bring them to the cell surface with it. So we start to see interactions with these ones here. So you put prolines here, you still interact. But when you put prolines over here, you no longer interact. So this really leads us to the idea that the correct substrate, what the protein's really recognizing is a nascent MHC with very broad allele specificity for a chaperoning function. And you do need an element of substructure that has to form for, there's a minimal something that needs to be recognized. It is a class 1 specific chaperone after all. It doesn't just engage anything. And then as that MHC matures and binds to a peptide, if you're an allele like HLA-A1, you start losing dynamic motions that may be important for exposing an excited conformation, that is the correct conformation for high affinity interactions with the chaperone. But if you are a different allele like HLA-A2, you have more dynamic motion even in the mature form, and you're still able to in those dynamic motions, expose the correct conformations for conformational selection. And that's important because not only is there a chaperoning function, but TAP-BPR can also engage folded MHCs later on in the pathway, and it can edit and modify the peptide repertoire that is displayed. I'm going to say more about that later. So it's really two mechanisms of action that TAP-BPR has. Okay, so back to the mechanism here. So this is what people believed in 2007, or some people, let's say, believed in 2017. This mechanism was proposed by the group of Robert Tompé, very prominent scientists in the ABC transporter and antigen presentation fields. And what they were claiming is that there is a loop from the chaperone, shown here in orange. They called it the scoop loop, where this scoop loop physically enters the peptide binding groove, and it binds and stabilizes that groove in an open conformation. And this scoop comes in, and it can literally, by physical displacement, it scoops out a peptide. And so if you had, say, a low affinity suboptimal peptide here in yellow, the chaperone will recognize it, maybe because it's a bit unstable here, and more dynamic motion, recognizes it, binds it, and scoops out the peptide. A new high affinity peptide comes in. There's physical competition, steric exclusion between this peptide coming in and the scoop loop, and that bumps off the chaperone. Versus more distant effects, conformational effects. So that was the idea. Very elegant, very simple, but is it too good to be true? So this is the evidence for that scoop loop, which at this stage, for all I know, might have even made it into immunology textbooks by now. The evidence was based on this structure here. This here is showing the, so the structure is shown as a ribbon, the electron density is shown as the mesh, and then the structure is colored by B factor. For people that aren't familiar with crystal structures, the B factor, when you build your model, the B factor is kind of like your error. It is a measure of the error in your ability to assign X, Y, Z coordinates to an atom. So the higher this B factor, the more uncertainty you have in that atom's position, possibly because of bad electron density, dynamic motions. There could be different reasons for that. So this was one structure from Tompé's group. They modeled in these two helices are the rim of the peptide binding groove. The peptide would be here, and they modeled in this scoop loop right here. This here was a second structure from the group of David Margulies, and they published back-to-back in Science, and here they didn't feel that they could see much density here. They never modeled anything in. Two structures had very similar resolutions, claiming to be similar quality, but you'll notice the one with the scoop loop modeled in, the B factors are very high, abnormally high, and the B factors for the scoop loop in particular, which we don't even like to call a scoop loop, so you'll see on some of these figures it's going to be called a loop based on its residue numbers, astronomically high B factors. You have to question, was this real? Tompé's group had another structure, a cryo-EM structure, published about a year later I think this one was in Nature, of the peptide loading complex, cryo-EM. This is now, those previous structures were of tapicin here, of TAP-BPR. This cryo-EM structure is now of tapicin. Here so again, these two helices here, those are the rim of the peptide binding groove. The peptide would be sitting right here, and they are partially modeling the scoop loop. They didn't model all of it, but they're partially modeling the scoop loop going into the cavity. But if you actually look at their electron density, there's perfectly good electron density up here that they never modeled into. There's no electron density where they're modeling a scoop loop. And if you look at the B factors here, they are a perfect 30.00 square angstroms, which for people that work with structures will know is impossible unless somebody refining the structure has deliberately excluded those residues from the refinement process. So that is the evidence for a scoop loop. So, you know, both Nick and I, coming from David Baker's lab, very proficient in Rosetta. So we did some Rosetta modeling, build a whole lot of different possible loops here, whole bunch of them. They're kind of wafting out in the breeze. They never, ever do we see them ever entering the peptide binding groove. So we never, in modeling at least, we never see any evidence of a scoop that goes into the groove. We did a experiment where we took cells and we knocked out tapicin, the critical chaperone in those cells. You knock it out, so now we've got all of our pieces here that are meant to come together, but they can't because they don't have the critical chaperone. They never make it to the cell surface. If you put tapicin back into these knockout cells, tapicin now properly assembles in the peptide loading complex. You properly load the MHCs. You rescue surface expression of MHC class 1. But we can also put in tap BPR. We modified it a little bit. We put on the C-terminus of tapicin to recruit it to the transporter that shuttles these peptides. It's, you know, tap BPR is not a perfect functional replacement for tapicin, but it's a partial replacement, and we can partially rescue surface class 1 expression. So we use this as the basis for a deep mutational scan, where now we could do a mutational scan on tap BPR and see what are the important residues for chaperoning and rescuing surface class 1 levels. You know, up here I've mapped data, you know, that's thousands of mutations mapped onto the regions of tap BPR that were included into the library. Orange means that that's a critically important site that is intolerant of mutations. And then pale colors would mean that it's an area that's tolerant of mutations. And what we see is that the most important regions, it's this little area here, which is buttressing the underside of the peptide binding groove. And then it's this region here, which is at the interface where beta-2 microglobulin needs to associate with the alpha-3 domain of the MHC heavy chain right here. Those are the critical areas. The scoop loop, the so-called scoop loop, would be right up here, and you can see that it is pale, it's white. That means it was totally tolerant of mutations. So it doesn't really matter if you change residues up in this scoop loop, which you would not expect if it was really entering and making explicit contacts inside of the binding groove. So in Nick's group, they labeled the MHC molecule with isotope labels on methyl groups, and then they add in tap BPR, and they look at the NMR spectrum, and then they throw in either wild-type tap BPR in red, or they throw in tap BPR where they deleted what is supposedly the last turn of this little helix here for you know, was modeled in the scoop loop model, delete that supposed last turn, or just delete the very tip of that loop, the terminal 2 residues. And what you would expect is that if this loop really is contacting the base of the binding groove, then you would see, as you delete that loop, you would see changes in the chemical environments for these isotope labels here, and you would therefore see changes in their chemical shifts. And there is none. There's no changes. It doesn't matter whether you delete that loop or not, it looks the same. Maybe there's contacts to a peptide. So here, this is much cleaner spectra because the labels are now on the peptide. Maybe there's contacts between this loop and the peptide, explicit contacts. So again, wild type, big deletion of the loop, small deletion of the loop, no change. So there is no evidence that there is any physical association between this loop, any explicit contacts between this loop and the peptide or the base of the groove. And then these are ITC measurements to look at a balanced thermodynamic cycle here, where we can do a whole bunch of ITC. We can look at each of the different steps here. And this is looking at the ratio of the different KDs here between a scoop loop deletion, that's the delta 2 here, and its ratio to wild type tap BPR. So where are the differences? The differences are first on KD1. KD1 is the affinity for tap BPR binding to empty MHC. And what we observe is that when you delete the scoop loop, the binding of tap BPR, the KD gets smaller, the binding's getting tighter. That is the complete opposite of the scoop loop model. The scoop loop model, if you're really making physical contacts between the loop and the base of the MHC, then as you delete that loop, you're losing atomic contacts, the KD should increase. It should get worse binding, but that's not what happens. It actually gets better. And then KD4 here, KD4 is the binding of the peptide to a chaperoned MHC. And what we see is that when you delete the scoop loop, the binding of the peptide, the KD for the peptide is getting bigger, it is binding weaker. Again, the exact opposite of a scoop loop model. In the scoop loop model, there is physical competition, exclusion between the loop and the peptide. But we see that you delete that loop, the peptide is actually getting in and binding. You delete the loop, the peptide is not held as well. So the loop is actually helping keep the peptide in the groove. It is wafting over the top of the groove, just as we modeled. It's kind of everywhere. That's why you don't see it in the electron density of those prior structures. And it's really a kinetic trap. And without going through the detail, that just makes total sense from the biology. Because from the biology, you've got these MHCs that are bound to a peptide. They've already been chaperoned, they've folded, and now you're at the quality control step, where if you re-engage, if you're a folded MHC with a peptide and you re-engage a chaperone, the chaperone only wants to transiently sample that peptide MHC, and if it is a tight peptide, you don't want your chaperone just to be scooping out any peptide willy-nilly. If it's a tight peptide, you want it to remain there. The groove is going to open, it's going to widen, it's going to be weaker affinity for the peptide, but by having this loop above as a trap, you are minimizing the dissociation of the peptide. And if you're a weak affinity peptide, well, that peptide trap isn't good enough. You're still going to dissociate, and now you can go back through the cycle to pick up a new and hopefully higher affinity peptide for presentation. So when you look at it afterwards, when we have all the data, the scoop loop model also just doesn't make sense for the biological, for the biology of what this protein has to do. Okay, so the final little story here on these chaperones is how we can engineer them. So I've shown you that tap BPR and tapicin can act with a classical chaperoning function, helping fold these MHCs, but then they can also bind to some, not all, but some MHC1 alleles, and when they do that, they can catalyze peptide exchange. So they can bump off weak affinity peptides and allow higher affinity peptides to come on. So this is how a chaperone like tap BPR may function as a peptide exchange catalyst. So your chaperone has some weak binding energy to a peptide MHC. We even know what that is. We can measure those binding affinities. There is a much tighter binding affinity between an empty MHC and the chaperone. So these are micromolar affinities. These are nanomolar affinities. So the chaperone is really stabilizing the empty MHC intermediate, and then another peptide could come along, and then the chaperone can release this alternative peptide MHC complex. So this looks just like you would expect for an enzyme. So you're lowering your activation energy barriers here to catalyze peptide exchange. The problem, And we would love to do this for certain applications. So I can, there are many in vitro applications where you make different peptide MHCs. They're used in diagnostics. There's other applications for them. You could also think of peptide vaccines where you could in vivo use some chaperone like this to facilitate exchange of peptides on endogenous MHC on cells to put in whatever peptide you want to have an immune response mounted to, a cancer neoantigen, for example. So the problem with that, though, is that TAP-BPR is only recognizing a small number of HLA-1 alleles. It recognizes HLA-A2, that's great, that's a very important allele, but HLA-A1 is also another very important allele, and there are some other ones you would like to also have this working on. So we wanted to broaden the substrate specificity for TAP-BPR interacting with folded MHCs. So we took two approaches to do this, and they were done independently in Nick Skarakis' lab and in my lab. First, I'm going to show you what Nick's lab did. They took the approach of looking at different homologs of TAP-BPR. They looked at different species here, and in particular, their attention was drawn to chicken. It's quite diverse from human TAP-BPR. Chickens are also very interesting in that the TAP-BPR in chickens on the chromosome, it travels in the same locus as your your MHC1 genes. So that means that MHC1 alleles, as they diversify in chickens, are co-evolving with their chaperone TAP-BPR, whereas in humans, TAP-BPR and the MHC1 loci are different, so they don't go together. So their attention was drawn to chicken. So here's in the surface, TAP-BPR is in green. Here's this is from a structure here. There's the MHC there. And if you look at the contact surface here, you take TAP-BPR, peel it away. The blue here are residues at the interface that are common between human and chicken TAP-BPR. The red are positions that differ between the two species. You can see there's a lot of differences. There's a lot of red here. So that was the approach they took. They did SPR measurements where they immobilized the chaperone TAP-BPR on the sensor surface, and then they came along with peptide MHC complexes. These tend to be weak micromolar affinity interactions. The peptide that they put on the MHC was photolabile, so they could UV irradiate it. That causes photolysis. The peptide breaks. You can now make an empty MHC in the experiment. It's unstable, so that's why you go through this effort. You can't purify this empty MHC, but you can generate it in situ. And now you can measure the affinity here between an empty MHC and TAP-BPR. This is nanomolar, tends to be nanomolar affinities. Looking at many different alleles here, you can see that some human alleles of MHC1 only interact with human TAP-BPR. Some human alleles of MHC1 only interact with the chicken protein, and some interact with both. This is looking at peptide loaded. When they look at peptide deficient here, in black are the alleles, the human HLA-1 alleles that interact with both chicken and human TAP-BPR. Blue means it only interacts with human. Red means it only interacts with chicken. So basically, you can pick up differences. And you can do in vitro peptide exchange experiments, where here you have a peptide loaded onto a MHC molecule, and then you come along with a fluorescently labeled peptide that you want to do exchange with, and you can look at fluorescence polarization and the rate of exchange here, and then you can add some amount of TAP-BPR as the catalyst. So in pale blue here is no TAP-BPR, no catalyst, so the exchange between the peptide bound to the MHC and whatever peptide you're adding into the solution is very, very slow. This is over hours here. But as you start adding increasing amounts of TAP-BPR, going to purple, green, red, and black, then you can now start to see very rapid exchange between the two peptides. So this is showing TAP-BPR functioning as an exchange catalyst in vitro. And we can look, here's, for example, one allele, HLA-A2. We can look at the apparent exchange rate of peptides. Human TAP-BPR in greenish blue here works really well for HLA-A2. The chicken TAP-BPR doesn't work so well, shown here in red. But now you look at another HLA-1 allele, and the chicken works really well, but the human doesn't work so well. So now, depending on what allele it is you want to catalyze exchange on, then you choose which homologue for the experiment. The approach that we took in my group was we decided to take a directed evolution approach and use deep mutational scanning, where we noticed that if you take TAP-BPR, so these are cells where they don't express tapicin, so they're knocked out for tapicin, no surface MHC, but they do have HLA-A2, they do have HLA-A1 alleles of the MHC. So if you put in TAP-BPR into these cells, you can, this is basically a measure of rescue here for the background levels that are leaking through. We can boost surface MHC. You put in some mutations. These are mutations that interfere with binding to beta-2 microglobulin. So if you are a folded MHC and you're properly folded, you are now have a heavy chain associated with beta-2 microglobulin. If you disrupt those interactions with these mutations, if anything, the TAP-BPR is actually working even better. That's because this is a chaperoning function. It doesn't care whether the MHC is properly folded with beta-2 microglobulin for this function. But if you put on that TM helix and you bring TAP-BPR to the cell surface, where now you can have interactions between folded MHC and TAP-BPR at the cell surface. So you're now bringing things to the cell surface. Now you put in these mutations here, identified as TN6 in the field, that disrupt beta-2 microglobulin interactions. Now, boom, you totally kill interactions at the cell surface. So this, when you bring TAP-BPR to the cell surface, we consider this a model for looking at interactions between folded proteins, which is akin to the peptide exchange mechanism. That's interactions, again, between folded proteins, not chaperoning. There's nothing for HLA-A1 because TAP-BPR just does not interact with HLA-A1. So we did deep mutagenesis. Orange, again, are bad mutations. Pale colors are neutral. There's not a lot of blue, but we're trying to find things that engage HLA-A2 better at the cell surface here. Data quality, this is for mammalian experiments, mammalian cell experiments. This is excellent reproducibility between independent experiments. The red here are premature stop codons. They should all be in the negative quadrant. They're deleterious. So good data quality here. And this is mapping that data to the surface. And the main takeaway from doing this was that we now saw this large orange patch, meaning these residues are mutationally intolerant and are very important. We saw this large orange patch now where you have interactions with beta-2 microglobulin indicating you're interacting with a folded MHC. That's shown here. This is a color difference between our previous scan, which was intracellular, chaperoning, to now our scan that is at the cell surface, peptide exchange, and the cyan is showing the gain in conservation for this second scan at the cell surface. So we're seeing a lot more conservation of residues that are interacting with regions that are only in a folded MHC substrate. So that was great, and then we tried to pick out from here, from all of this data, we tried to pull out mutations that are unique in improving interactions between the chaperone TAP-BPR and folded MHC at the cell surface. not for increasing interactions inside the cell, but really for folded molecules. And we start putting some of these mutations in, and we see that indeed, when you put in these mutations in the cell-based assay, we are increasing surface HLA-A2 as we engage it at the cell surface and keep it there. In the in vitro peptide exchange assay, these mutants here, so the wild type is in this pale blue here, and these various mutants are much better exchange catalysts on different HLA-A2. I'll point out this here is a log scale, so the exchange rate is increased about 100-fold. And then the real kicker was that even though all of our selections were based on HLA-A2, by increasing interactions to this allele, we now were able to interact. Wild-type TAP-BPR has nothing for A1, but now we can gain interactions with this alternative allele. This is, again, the cell-based data, and then this is in vitro peptide exchange. Pale blue, again, wild-type TAP-BPR, zip, nothing. And we put in different mutants. They work really well. Chicken TAP-BPR also works well. And then when we got together and we compared all of our data between our labs, it just so happened that they converged. So mutations, I won't go through the details here, but mutations we were picking up to some of these residues when we looked at it, totally unintended, but we were picking up mutations that are either in tapesin or in chicken TAP-BPR, and we ended up putting those mutations into the human TAP-BPR protein and altering and broadening its allele specificity. Okay, so yeah, so we're doing it for biotech applications. So very quickly, this is just my one slide for some work that I'm now doing in my company where we still think about HLAs and antigen presentation. This is a little different. We are interested in de-immunizing proteins using computational methods, where one of the approaches that we use is we can, so ordinarily if a protein drug, so this would be an antibody or an enzyme for some enzyme replacement therapy, you inject that into the human body, into the patient, it gets processed by antigen-presenting cells. Peptide antigens now from that protein get displayed. This is now class 2 MHC instead of class 1, but the principles are the same, and then it gets presented to a T cell. That T cell gets stimulated, and then this T cell goes on to release signals that will drive B cells in the immune system to make antibodies, anti-drug antibodies to your protein here that was being delivered as a drug. This is a major problem in the development of biologic drugs. Many biologic drugs, they have amazing specificities. They are often amazingly efficacious, but then they can fail in the clinic for immunogenicity reasons or manufacturability reasons. So what we can do is we can use computational methods to identify what those peptide epitopes likely are, and then we try to mutate them to reduce their affinity to MHC molecules while preserving the folding and the function of that protein that's being used as a drug. So this is some of the stuff that I now do in the company at Cyrus Biotechnology. Okay, so a bunch of people all contributed to this work. In particular, in my lab, most of the work was from Christine Devlin, and then a lot of the work I showed. Amazing collaboration we've had now between Nick's group at Children's Hospital of Philadelphia, and then for people that are interested, I gave you snippets of some of the data. If you're interested in looking any of it up, these are the manuscripts for the published work. And I'm happy to take questions. Yeah.
I guess I was curious about one of the initial slides you said, each HLA allele can interact with 10 to the 4 different antigens or thereabouts, right? So presumably there's a broad binding affinity range within that set.
Yes.
Right. So how does it decide? Does a weak affinity necessarily mean it's less immunogenic? Is that what it means? So how does it decide what is worthy of presentation and what needs to be left out or left in?
So in the beginning, you have, can go back to, I guess here's a good example. So in the beginning, you have this peptide loading complex, and you have the transporter here that's shuttling these peptides in. So you have a very high local concentration of peptides around the MHC. And you can get many of these peptides will go in, and some of them are not going to be the best peptides. And then the problem with that is that as you go to the cell surface and then reach the cell surface, those peptides can fall off. And when they fall off, well, that's a useless MHC, and at some point it will shuttle back into the cell to pick up a new peptide. So you don't want to just load up your MHCs with weak peptides. And this is where the primary role of TAP-BPR probably comes in. It's probably more of a quality control step, where it's really working mostly in the Golgi, where these peptide MHC complexes are migrating through the secretory pathway. And if they have a high affinity interaction with TAP-BPR in the Golgi, as TAP-BPR engages them in the Golgi, it's weakening that affinity for the peptide. And if you're a weak affinity peptide to begin with, you come off, and now you can get deglycosylated and driven back to the ER. But if it's a high affinity peptide, then you weaken it, but you've got your loop sitting on top. It doesn't just come off, and you're going to have your equilibrium, and as the groove closes back up in its conformational equilibrium, it has lower affinity for the chaperone. The chaperone just falls back off, and onwards you go back to the surface. But you're absolutely right. There is a huge spectrum, and this comes up in the computational models to predict affinities of different peptides. It's not like you're a binder or you're not a binder, and you have to choose some thresholds along the way. Do we say we're going to take you if you're in the top 5% of affinities, top 10%? Obviously, if you're in the bottom 50%, I think we can exclude you as being biologically relevant. But yeah, there's a huge range. But most biologically relevant epitopes will bind in around the nanomolar KD. Yeah.
While we're on this slide, what is the function of this UGGT1? Is it functionally important?
Yes, this here is a glucosyl transferase. So these chaperones form complexes. So I showed you tapicins forming a complex with these housekeeping chaperones, ERP57, calreticulin. They're involved in many different proteins. That's in the ER. TAP-BPR is ordinarily it would be in the Golgi. Here we just relocalized it by changing part of the protein.
So it's mislocalized.
We relocalized it to the transporter by putting on this segment of tapicin. So this diagram for this assay is a chimera of TAP-BPR ectodomain with the tapicin transmembrane cytoplasmic tail, because we wanted to localize it to the ER with the transporter. So that's what this is. But ordinarily, TAP-BPR would be in the Golgi, and it interacts not with ERP57 or calreticulin. It interacts with this glucosyl transferase, because the glucosylation of the MHC is also important for its biology, and putting on glycans or taking them off can drive a bad MHC back to the ER.
Two more questions.
Yep.
What is the loss of function of TAP-BPR? Is it phenocopying the tapicin loss of function?
So we use it as a phenocopy of tapicin. That's how we use it, and we use TAP-BPR just because we can make it. It works great in NMR. We can make bucket loads of it. Tapicin, we are just starting to make some good amounts with some engineering of it, but much harder to work with. But it's not strictly a phenocopy. It has, tapicin's pretty much essential if you want a good functioning immune system. TAP-BPR, it's not. It's not essential.
So probably less MHC class 1 on the plasma membrane as well?
It's not less if you knock out TAP-BPR, but there are subtleties in the peptide repertoire that are presented. So it is modulating which peptides are going through the quality control.
Also, in terms of the exhaustion, inducing the exhaustion, can you actually manipulate the system to have less or inefficient antigen presentation, or even having less MHC class 1 on the plasma membrane in a certain time frame?
I've never thought about that. I would think it's possible if you had a way to manipulate the expression levels. And obviously, you know, there are cases where if you know enough about the pathways that drive expression of your protein, there may be small molecules that you can add to a system that dampen it. So if, I don't know, but if such, you know, molecules exist to modulate its expression, I don't see why it wouldn't be possible. Sorry.
Hey, great talk. I really liked the deep dive on thinking about the loading of the peptides. Towards what you were saying at the end about the kind of entrepreneurial direction, if I had a synthetic protein that I wanted to fly under the radar, would you think it's better to make it invisible in the sense of the peptides that are produced from it are bad for loading into MHCs to try to keep it as low a profile that way? Or I've seen other strategies where people try to build the protein kind of out of fragments that are all things that, you know, are self-derived. And I'm just curious if you have any kind of deeper thoughts maybe on design strategies for visibility.
The more human-like you are, then there's the idea that the less, and sorry, my voice just went dry, the less immunogenic you will be. I can give a very specific example. Camelid nanobodies. You know, they're kind of still all the rage. They were very much the rage a few years back. One of those has reached the clinic. It's great, barely immunogenic at all, and they humanized it. So, you know, so that's one strategy. Humanize your protein as much as possible, and then hope you've humanized all the key epitopes so that the immune system is blind to it. The other strategy would be make it so it's not presented. And that can mean different things. That can mean, like, you know, what the company's doing on complex proteins where we're trying to prevent their presentation on MHC. For some mini proteins, mini design proteins from the Rosetta design community, they like to think, it's not, you know, none of this has been proven in humans yet because you've got to get to the clinic to really prove it. But, you know, there is some in vivo data that is suggestive that if you are super stable, hyper stable, you're not well processed by the proteases necessary after you go into an endosome to cleave you up and put you onto a class 2 MHC. And therefore you get poor presentation to CD4 positive T cells, and you're thus not very immunogenic, and you don't elicit antibodies. There's probably some truth in that. So there are different strategies. I think all of them could be beneficial, and all of them could help depending on your scenario. Yeah.
Have you looked at MHC alleles that are associated with autoimmunity to see how they interact with tapicin or any of these proteins?
Not explicitly, as in we haven't like looked to make anything of that, but I can tell you that some of the alleles in the sets that we have are absolutely associated with certain, you know, autoimmune disorders. But we haven't gone to tease those ones out to say something specific about them. But there are some in the proteins we've examined. To go further, in Nick's group, I didn't show the data because obviously I was biased towards showing mostly data from my own group. But in his group, in the recent publication we just had in Science Advances, they had a bead-based assay, and they screened, I don't know, like 100 different alleles of different MHCs. So it's pretty exhaustive, and all the important ones are in there that are most prevalent in the human population. So it's all there, the data's all there. Someone just needs to look at it with that question. Yeah.
All right. Well, let's thank Erik for a great talk. Thank you. Thank you.